So welcome everyone. My name is Fritz Sedlazeck. I'm an associate professor at Baylor College of Medicine, as well as an adjunct associate at Rice University. And so today it's my great pleasure to introduce Read2Tree, which is a new method that Christoph Desimos' lab and my group has developed over the past years to exactly what was just introduced, to utilize raw reads directly to place and infer phylogenetic trees. So when we think about comparative genomics, the current status is often that we first of all assemble the species or the organism that we're interested in. So we just recently did that with the T2T consortium together to resolve a human genome. And one of the recommendations of this consortium is nowadays to utilize 55x Illumina, 55x nanopore, and 55x bug biosequencing, which is of course quite expensive to do. But in any ways, if you're trying to achieve the whole T2T, or if you're just achieving to get a really robust assembly together, it's quite costly, and there are multiple hurdles. So besides the large sequencing requirement, of course, you also require a large compute infrastructure, such as cloud computing or local clusters. And then you require expertise to not just close the gaps, but also spot errors, or for example, rearrangements in your assemblies, but also contaminations from other sources. The next step is then typically an error correction, which is often required in an orthogonal sequencing step, for example, Illumina. And then also you have to have expertise to kind of polish and how often you polish your genome, because over polishing your genome will lead to eradicating of heterozygous locations of your assembly and introduce furthermore errors or eradicating repeats on their own. And then as a third step, and that's really one limitation step that is currently heavily impacting consortia, is the annotation step, where we have multiple tools such as Makers or Breaker that deliver very different results, quite frankly. So it's often recommended to do RNA sequencing on top to improve the annotation of your genome of interest. And then always you require manual correction annotation that requires further expertise. So then finally you get into a homology interference between the genes that are actually either all against all, which is often quadratic, and so for larger organisms, as you probably better know than I do, is quite computational intense, or you utilize an HMM profile approach. And then you can go into the multiple sequence alignment and then the tree interference of your choice, like U-tree, fast tree, or whatever. So with all these pipelines and all these steps, it's pretty clear that there are multiple challenges for small labs or medium-sized labs to achieve the sequencing coverage, but also the expertise. But it's also a challenge for these large consortiums, such as the EarthBio project that I'm also a part of, as well as the Vertebrate Genome project. And most of them are actually currently stuck not in the assembly because they are well funded and can produce the sequencing data, but they're really stuck in the annotation part. This annotation part is currently really the bottleneck of all of these things. And so Christoph and I were thinking about how can we overcome this. And so what I'm going to present today is basically this Read2Tree approach that not just is a pipeline across these steps, but basically it's a pipeline to avoid many of these steps and more streamline your approach down to the tree inference, where you can then use whatever method you fancy, so to speak. So how does Read2Tree work? Well, first of all, what we are doing, and that's one of the main tricks behind the program, is we take the raw reads, which can be DNA, RNA sequencing, it can be long reads, it can be short reads, and we map them directly onto orthologous groups obtained from the OMA database. So the OMA database is an orthology database maintained by Christoph's group. Then we construct pair-ortholog consensus sequence based on your species or based on your reads. We select consensus sequences based on the mapping itself, the coverage that we obtain, and the sequence length that the consensus sequence stretches. So there is a pre-selection process in place. And then we do something which I call a slim MSA. It's probably not the right term, but basically we already know from these orthologous groups the multiple sequence alignments, right? So the only thing we have to add is one more sequence that we just reconstructed in a consensus sequence. And we know how this looks like because we just constructed it based on a more close related sequence. So the first thing that we of course want to see is like how good or how bad is Read2Tree, right? Because it's a really different approach to what is out there right now. So for this, Sina simulated a data set. It's pretty straightforward, 15 genomes containing 100 genes just to see if it works, right? We simulated 15 different branch lengths. We simulated Illumina reads from coverage values of 0.1 up to 10x coverage, so just testing out the extreme cases, right? And so we looked at the performance, and you see these plots on the right, where on the y-axis is the Robinson force distance to the simulated tree, and on the x-axis are the BEM, so the branch length difference, or the branch length itself. And you can see at 0.1x coverage, which is basically nothing, right? The method struggles a little bit, but then when we increase coverage to 0.2x, to 0.5x, 1x, 5x, and 10x coverage, right? We see basically that the Robinson force distance is 0. And when I can draw your attention also on the x-axis, we simulated quite an evolutionary distance, right? So like here in the middle, I hope you can see that, you have the BEM value of 100 up to 140, and 100, just to remind you, means that on average, there is one substitution per site, right? So like the evolutionary distance, just in the simulation, is pretty high, and we can still achieve accurate results, even with very, very low read coverage from Illumina reads. So of course, this is simulation. So like we wanted to test this in real case, right? So we utilized the data sets from Short Read Archive, obtained data from Arabidopsis thaliana, DNA data. We downloaded mus musculatus data, RNA sequencing data. We downloaded yeast data from, again, DNA data. And then we started to kind of delete basically delete different closest ancestors, right? To kind of really simulate this reference distance, because our method, as I just described, is really based on mapping against this orthologous group. So we wanted to see if it breaks down, right? So as you can see here, in case of Arabidopsis, we simulated up to 117 millions of years of evolutionary distances by kind of not providing these closest ancestor sequences. For mouse musculus, up to 600 years, and for yeast, even up to 1.1 billion years, which is an astonishing evolutionary distance, I think. So the first thing that we looked at with these different data sets is how good is our consensus sequence in terms of precision, so how well does it recover our sequence from the original species, and in terms of recall, meaning how many genes can we recover. So the point where you want to be in this plot is on the top right for all of these data sets. As you can see also on the y-axis, we have different y-axis. You have Illumina data on the top, nanopore data in the middle, and then bio data on the bottom. So testing all different technologies in one go. And so what we learned from this is basically that the recall is, of course, impacted by the coverage that we have, right? If we have really low coverage, well, some of the genes we will never observe, right? That's pretty obvious. The precision remains really, really high in terms of the consensus sequence on the y-axis, and this is the really good news here. Also, it's pretty robust, as you can see, across DNA versus RNA sequencing. In RNA-seq, we see a little bit of better results. We think that's because you have different transcription rates of the transcripts, and so therefore more coverage than the average genome coverage in some of these cases, right? And then, as I said, like across the sequencing technologies, you see it actually works also really, really well. So next, we were interested in tree accuracy, where we also measured the precision and recall, similar plot. So you want to be on the top right of every of these plots. And what we can see is that the precision in general works really well. The recall is, of course, again, impacted by the coverage. So, because like if you don't have the consensus sequence, you cannot have the tree recall. The only exception for the precision, however, is the Cerevisia data set, right? But like, as Christoph always tells me, yeast tree is always poorly resolved and leads to issues and with huge divergence times anyway. So maybe that's what we are also seeing here as an artifact of the performance measurement of Read2Tree. So as you can see, and I think we show over these two simulation as well as real data across different species a little bit and across different technologies, Read2Tree really performs great in terms of robustness of coverage. I forgot to tell you that. So the different colors in these plots are actually representing the different deletions of the closest ancestors, as indicated in the top tree. And then the symbols are actually representing sequencing coverage itself. So we really went from 0.2x sequencing coverage up to 20x sequencing coverage. And it's pretty robust and pretty remarkable. So we were actually also surprised that it works that well. But of course, the goal here is to kind of enable a better workflow, enable a different workflow than assembly would give you, right? So the next step is, of course, to compare these data sets and compare the performance with assemblies itself. So for that, for our Rhodopsis and yeast, our DNA data sets, we utilized megahit and SubDenovo for assembling these Illumina data sets. And then for long reads, BacBio and Nanopore, we used Canoe itself, which is a pretty robust assembler that I think everyone knows. For RNA sequencing, we only focused on the assembly of short reads with Trinity itself, and didn't do an assembly of the long read RNA sequencing. And of course, we measured the runtime and the accuracy. So how you read this plot is again like on the y-axis, see the difference in accuracy between Read2Tree and the assembly, annotation, and correction approach. And on the x-axis, we this time plot the distance in millions of years when we do the same thing with these data sets as the before plots. And so you see the line indicated 0.0, which is basically the line that distinguishes if the assembly is better, which is the top above the line, and where Read2Tree performs better. And so what we can see across the species and across the technologies is that especially with low coverage data sets, Read2Tree definitely significantly performs better because the assembly is a little bit hindered to complete. And then only in the case of yeast with higher coverage and really high divergence rates, so again, this is up to 1 billion years of difference in the reference, we see a performance improvement of the assembly itself compared to a Read2Tree approach. Nevertheless, when we measure the runtime, we see, of course, and what we assumed is that Read2Tree is orders of magnitude faster than the assembly and the annotation approach, right? So the assembly here is indicated in red, and in blue is Read2Tree. And you see here on the x-axis hours and time in hours on the log scale. So with this kind of method at hand, right, we really kind of looked out and see that it's quite flexible. And so we were really interested in leveraging some of the short read archive data, because this has the potential to really help us to construct larger phylogenies and get more insight in evolutionary distance and relationships across different species. So the first thing that we actually settled on was the following data sets. So Shen et al. Published a really fascinating paper in Cell 2018, where they built the phylogeny of yeast. You can see the phylogeny on the right side. And so actually we took all the data sets and even added data sets. So in total, we run Read2Tree on 435 yeast genomes in just a fraction of time. And you see that the two trees are very similar to each other. You see there's a little branch, for example, on the far left side that isn't represented in the Shen et al. Tree. As far as I recall, this has really, really low coverage of 0.1x, so that's why maybe it's replaced. But in total, the trees are really concordant to each other, whereas the Shen et al. Group took like months to years of compute and efforts across multiple groups to accomplish these data sets in terms of assemblies and annotation and tree building. The next thing that we wanted to apply Read2Tree is, of course, coronavirus, because we have been all interested and impacted by this one bastard, so to speak. So here we challenged Read2Tree, right? Because we wanted to see how it performs in SARS-CoV-2, which impacts us, but also across other coronaviruses. So we submitted the entire data set, just the raw reads from SRA directly, to Read2Tree and computed this tree. And the most fascinating thing, I think, in our discussions came out that not just is the tree correct on the broad width of spectrum, right, across the different coronaviruses, but it also resolved automatically the SARS-CoV-2 tree accurately in terms of delta and alpha and omicron data sets. And this really indicated for me that we can use Read2Tree, of course, for doing the phylogeny, as I just told you about, but also for future experiments when another pathogen is coming around, we can sequence that really rapidly with low coverage and see where it's going to be falling into the phylogeny of different viruses, which is always very hard to do. To top this, of course, one of the reviewers in our submission asked for a larger tree, which is, I guess, always the case. They always want to see a larger tree. And we currently actually finished processing a COVID tree of 10,000 10,000 data sets, 10,000 samples of it. And the most challenge of that was honestly finding the right samples in the RSA and getting them correctly annotated. Christoph's group was basically able to do this 10,000 COVID tree data set within like a week or two with their cluster. So that was really straightforward. And it shows again like the scalability of Read2Tree. So to summarize a little bit, what is Read2Tree? Well, Read2Tree is really a rapid phylogenetic tree construction based on raw reads. It scales, I think I showed you that, from low coverage, so we showed you 0.2x up to 20x, to large numbers of samples, such as, for example, this 10,000 COVID tree. It also eases the comparative genomics from small to large labs. I think this is really a major point I want to stress, because I've seen so many different smaller labs that really struggle to produce 55x of BugBio, for example, coverage. It's really costly, and you often don't get funding for exactly that. But also for larger labs. So as I said, the EarthBio project is really something that we are also in communication with, as well as this Sanger Institute with their current Tree of Life project, I think that's the name, are also looking into Read2Tree as just an orthogonal pipeline to their assembly pipeline to see if the concordance is there, or if something went wrong in Read2Tree or something went wrong in their assembly pipeline if the concordance is not there. So some of the future directions that we are currently thinking of, as I said, like I wrote it down here, a larger COVID tree, because the reviewer wanted to have that. This is basically done. We are currently playing around with how to visualize this tree. But then what we really want to dig into as well are metagenomic samples, so where multiple species are present in one sequencing go. And this is basically a task where we have to dig in a little bit more on the creation of the consensus sequence. So we will basically work on clustering the reads all together for each of the orthologous groups, and then split them up in different species that we see from the haplotype clustering. And the other intriguing thing that I think is really cool to think about is single-cell genomics or single-cell metagenomics, because Read2Tree can work on this 0.1x, 0.2x coverage. That is often what we gain from single cell. We could potentially utilize Read2Tree really in digging into these data sets and really characterizing a whole environment altogether, very, very detailed. And so, yeah, if you're interested, there's much more information in our preprint that is up on bioRxiv, so please check it out. So before I'm ending this talk, I wanted to shout out two things, though. A little bit selfish advertisement here. So the first thing is we are organizing a hackathon again at Baylor College of Medicine. This is going to be the 4th hackathon that I'm organizing. And so like everyone that is interested, please consider applying to contribute to this. On the top right, you see the roadmap of all the participants from last year's hackathon. So we really try to be inclusive here and really incorporate multiple people really from around the world. Usually we end up about 90 or 100 participants, and we publish a F1000 paper as a report for each year with all the participants. So down here is the third hackathon that we published this year in F1000. And so in 2022, this year, we are planning the event to be in October 9th to 12th, with support of DNA Nexus, BugBio, like Oxford Nanopore, NCBI, and NVIDIA. All these gives us support of different funding, as well as prizes for the best groups. Topics are a wide range of virus databases, structure variants, because that's always my main topic, actually, and visualization of variants. And the last thing, I think I'm still a little bit in time, is my group is looking for postdocs and staff scientists. So like if you're interested, if you know people, please reach out to me. We require people in the analysis side, as well as in the development side of methods, especially focusing on human genomics and human genetics with Oxford Nanopore and Promethean and BugBio. And the Texas Medical Center is really a vibrant place with lots of funding, lots of opportunities in medical records. So closing up, I want to thank, of course, David, who was the main driver behind Read2Tree. Adrian and Sina really helped a lot along the way, especially on the COVID case and the simulations. And then, of course, Christoph, which was another great collaboration with you to kind of develop this tool. With that, of course, multiple universities such as UNIL and Swiss Institute of Bioinformatics, Baylor College Medicine, and Swiss National Science Foundation for funding. And I'm happy to take any questions. Thank you.
We have time for a quick question. Anybody?
Well, a quick question then. Hi, thank you very much for the presentation. So my question is about the comparison that you showed for the reconstruction of the yeast tree.
Yeah.
So it is nice that both topology are almost the same. My question is about the differences that you found between the two trees. So were you able to know which one of the topologies were the right one? Are you able to solve some parts of the tree, for instance, for which the Sharon and Al paper wasn't able to solve?
Yeah, that's a great question, and it's sadly a gray zone of my knowledge because I'm much more on the sequencing and the processing of the sequence read side. I think the short answer to that is we are still looking into that. We utilize the NCBI taxonomy to label the tree further. That's not that picture, obviously. And we are looking into what this little branch, for example, on the left is, if that's just a coverage artifact, or if it's a real biological signal. Yeah.
Thank you.
You're welcome.
I think we could fit one quick one in.
Have you tested a case where you have different sequence depths for different samples? Because
this is the case. So like when you look at the green bar, sorry, I didn't say that. The green bar represents a little bit the sequence coverage across the different samples. So you see some of the samples are really, really low in sequencing coverage, like 0. something x, and some samples are really resolved to 30x coverage.
Yes, because you build the consensus at first. So with more reads, you get more polymorphism than the ones that with lower.
Well, no, not necessarily. So with more reads, we obtain more consensus sequences across orthology or phylogenetic important genes, right? So like our placement becomes more and more robust, obviously, because we cover more genes. But as you could see also in the experiment that we did with the performance, the precision is pretty high. It doesn't matter what the coverage is. It's only the number of genes that we can recover and utilize for displacements is impacted directly by the coverage.